for introduction. Thanks everybody for your coming. So I'm gonna talk this morning about the microbial killing capacity of the most popular 3 disinfectant available in the market on various surface contaminating with high load of Salmonella. So in 1996, the United States government estimated the cost of medical and productivity of bacterial food illness, and they found that this type of illness cost the government every year around 2.9 to 6.7 billion dollars. The pathogen on the top of this list is the Salmonella species. So the Salmonella infection costs the United States government every year around 0.6 to 3.5 billion dollars. Based on CDC report, they found that around 1.4 million persons infected with Salmonella every year in the United States. 400 persons gonna die from Salmonella infection in the United States every year. Interesting to know that around 49 of Salmonella infection in human is due to direct or indirect contact with animal or animal products. In the animal side, in the same year, the United States Department of Agriculture estimated the prevalence of the animal shedding Salmonella, and they found that 5.4% of the United States dairy cows shed Salmonella species, and around 27.5% of the herd have at least one animal shed Salmonella species, and 20% of all culled cows shed Salmonella species. So the key connector in this round trip between animal and human salmonellosis is the fecal waste. So fecal waste is considered the main source for Salmonella in the environment and food chain. So the animal shed around 100 to 10,000 Salmonella per gram feces every day. And the animal need around more than 8 log of Salmonella microorganism to infect it. In human, the person need around 5 log of Salmonella to be infected. Therefore, several studies suggested that the 5 log reduction is a safe level for farm biosecurity and the prevention of Salmonella infection in both human and animal. So in this study, I'm gonna talk about 3 disinfectants that are popularly used in the dairy industry. The first disinfectant that I'm gonna talk about is sodium hypochlorite or chlorine bleach, as they named in the practice. So chlorine bleach is used in dairy industry since around 1846, so around 2 centuries ago, and it's used extensively in United States dairy industry. The oxidative power of the sodium hypochlorite comes from formations of hypochlorous acid with oxidative potential around 1.38. This oxidative power enough to kill the hazard bacteria, including Salmonella and E. coli. The second disinfectant that I'm gonna talk about is peracetic acid. Peracetic acid is a byproduct of catalyzing reaction between acetic acid and hydrogen peroxide. The oxidative power of peracetic acid is around 1.87, which is higher than the hypochlorous acid with around 30 to 40%. Right now, peracetic acid is the most common disinfectant using in the poultry processing plant. And the interesting thing about the peracetic acid is that the peracetic acid is working in wide range of temperature. Peracetic acid is considered environmentally friendly because its breakdown produce acetic acid and water and oxygen. Also, the most important and the biggest advantage of peracetic acid is its oxidative power relatively not affected by the high organic loads in the sample or on the surface. So right now, the dairy industry in need to disinfectant with a specific character. These characters are this disinfectant should be safe, should be powerful, and should have a short half-life. The only disinfectant present in the market that fulfill all these characters is the ozone. So ozone is the most powerful oxidant and safe disinfectant available right now in the market. And to generate the ozone, it's a very simple, straightforward process, just applying high electric current to oxygen molecule, and with this electric current, they can split the oxygen molecule to two oxygen atom. So the weapon of the ozone is the unstable oxygen atom. This unstable oxygen atom attached to another ozone molecule with weak bond, and when ozone molecule start to contact or attack the organic matter, this active or unstable oxygen atom released and start to destroy the microorganism. They destroy all the component of microorganisms, starting from cell wall, cell membrane, all the DNA component of the cell. But so as anything in our world, so it has advantage and disadvantage. The biggest disadvantage of the ozone is it's still not easy technology. It's complex technology, and it's not storable. Every time you want to use ozone, you have to generate it, and it's relatively cost compared to other chemical disinfectant. From this information, so I forgot to mention that the oxidative potential of ozone is around 2.07, which around double the oxidative power of the chlorine, bleach-based disinfectant, and the half-life of aqueous ozone or ozonated water is around 20 minutes only, and after that they convert it to oxygen. So it's 100% safe. From this information about the oxidative power of these three disinfectants, we hypothesized that the higher concentration of peracetic acid and the lower concentration of ozone or ozonated water, aqueous ozone, gonna provide efficient mean to reduce the salmonella contamination on different surface to a safe level. Consequently, our objective was to evaluate the decontamination power of these three disinfectants on different surfaces. We used 4 surfaces. We used plastic, nylon, rubber, and wood as an example to different degree of complicity surface that available in each dairy farm worldwide. And we need to evaluate this decontamination power of these three disinfectants on this surface that uploaded or inoculated with dairy cattle feces that loaded with high load of salmonella. So regarding to the experimental design, in crossover design, we used 14 strips from each material. These strips are soaked in dairy feces inoculated with around 6 log to 8 log of salmonella. And after that, we transferred these strips from the inoculated feces and hang it on a rack to dry for another 60 minutes. And after that, each strips was transferred to a sterile bag, and the treatment strips was treated or exposed to the 3 disinfectant. We used the concentration 200 part per million for sodium hypochlorite. We used 400 part per million for peracetic acid, and we used 9 part per million for aqueous ozone for 4 minutes exposure. And the concentration that we used in this study based on earlier study, and also this is the most popular concentration used in the practice. After that, we swabbed the surface and the culture on the Petri film rapid aerobic count plate and incubated for 24 hours at 37 degrees Celsius, and then we count the colony or the cells using the Petri film automatic cell counter. So the reduction rate and reduction factor and the number of colony and the percentage of killage was calculated and corrected using equation developed by ASTM, American Standard Test Material. The difference in the reduction rate between the three disinfectant was evaluated or was tested using Mann-Whitney test and Kruskal-Wallis statistical test. We used univariate linear regression to test the effect of the substrate or the effect of the surface roughness or complicity on the reduction rate of the three disinfectant. Also, we used the linguistic regression model to characterize the relationship or the ability or the microbial ability to reduce the Salmonella load to a safe level, which is 5 log. B less than 0.05 was used as a level for statistical significance in this study. So regarding to the reduction rate for each disinfectant on each material, so this is violent blot. It's a combination of box blot and density blot. So on the plastic, the three disinfectants were able to clear the surface from Salmonella. They were able to clear around 6 to 8 log of Salmonella on the plastic surface. However, in the nylon surface, sodium hypochlorite and peracetic acid were able to clear the surface. They decontaminate, they remove, destroy all the Salmonella on the nylon surface. However, ozone was able only to reduce the Salmonella load to around 5.6, which is higher than the safe level. So ozone also reduced the Salmonella load to a safe level on nylon surface. On rubber surface, the peracetic acid only, the disinfectant that was able to clear the rubber surface from all Salmonella. They kill around million Salmonella per milli on the rubber surface, followed by ozone and then sodium hypochlorite. In the wood surface, unfortunately, the three disinfectants were not able, failed to reduce the Salmonella level, at least failed to reduce the Salmonella level to a safe level. And this has led us to test the effect of the complicity of material on the reduction rate of the three disinfectant, and we found that the type of material has a significant impact on the reduction rate or on the microbial killing capacity of the disinfectant. And as you can see, the sodium hypochlorite, the type of the material, has a significant impact on the reduction rate of the disinfectant. And the ozone also, the type of the material or the complicity of the material, has a significant impact on its microbial killing capacity. Interesting, peracetic acid didn't show any didn't affected by the type of the surface. So they kill most of the bacteria on different level of complicity of the surface. To summarize the microbial killing capacity of disinfectant, we created this bubble plot. So the external circle indicated to the Salmonella load, and the internal circle indicated to the reduction rate. The blue color indicated that the disinfectant able to reduce the Salmonella level higher than the safe level or higher than 5 log. And you can see, as we mentioned, the three disinfectants able to reduce the Salmonella load to a safe level on plastic or on a smooth surface and nylon, as example to moderate rough surface. However, in the rubber, the only disinfectant was able to clear the surface from the Salmonella is the peracetic acid, followed by ozone, and the lowest microbial capacity was for the sodium hypochlorite. On the wood on the wood, so the 3 disinfectants fail to reduce the salmonella load to a safe level. On the right side of the slide, the probability plot created from the logistic regression, and this is showing the proportion of each disinfectant for achieving 5 log reduction in salmonella contaminating different surface. For example, you can see here that the ozone, 80% ozone gonna be decontaminate or kill salmonella with level more than 5 log. With this, so we conclude that the 3 disinfectants are excellent, working very nice on the smooth surface. However, with increasing the complexity of the surface, the microbial killing capacity start to change from disinfectant to another. So we have to figure out each disinfectant and what is the microbial capacity of this disinfectant on each surface. For example, as we mentioned on the rubber, so the peracetic acid is excellent disinfectant. You can use it for peracetic for the rubber, and it decontaminates all the salmonella on the rubber surface, followed by ozone. Ozone can make a reduction factor 50%, around 20% higher than the sodium hypochlorite. In the wood surface, in the complex surface, so one washing cycle not enough to decontaminate the salmonella. You have to, using sequential washing, you have to use more than washing cycle for the complex surface, using whatever the disinfectant that you're gonna use, but one washing cycle not enough to decontaminate the surface. And finally, despite the peracetic acid showed the highest decontamination power on the different surface with different level of complexity, but still the low production of harmful residual, making the ozone an attractive alternative disinfection for improving farm hygiene and biosecurity. So this means, so I think we right now working on the new study to increase the concentration of ozone, because this is the maximum concentration that we can get from the unit that we have in our lab. But I think increasing the ozone concentration more than 10 parts per million, I think it gonna achieve the decontamination power similar to peracetic acid, or increase, or it gonna be more efficient than the peracetic acid if you increase the ozone concentration.
Thank you very much for your presentation.
So, yeah. With that, I'd like to thank you for your attention, and I will be more than happy to receive any question or comments if you have.
I think if we can have some questions at the end of the session for our speaker. So thanks for your attention, and I'd like to call the next speaker, Dr. Liz Creswell. We thank our speaker.